Hi there. This is a a follow-on video from an earlier video I did on the initial stages of respiration, and this video is designed essentially to cover the stages after the link reaction, after the Krebs cycle. Probably starting with the electron transport chain, and then looking at anaerobic respiration, and if possible, looking at alternative respiratory substrates. So, essentially, as I said in the previous, in all my previous videos, you would need to have a set of my notes in front of you and a question booklet, all of which I can supply if you get in touch with me. You need to have those in front of you because I'm going to make a constant reference to diagrams and questions in these booklets. So just from the previous video, this page, which is on the back page, essentially the back page of my notes, is a very important page because it sets out an overview of respiration, and it shows you essentially you start off with one glucose molecule, and the one glucose molecule then is converted into two pyruvate molecules. So you start with one glucose molecule and just call it G, glucose molecule. That glucose molecule in glycolysis eventually becomes two pyruvate molecules, and just call pyruvate. And each of those pyruvate molecules then is converted to acetyl-CoA, acetyl-CoA. Each of those convert to acetyl-CoA, CYL-CoA. And then each acetyl-CoA feeds into the Krebs cycle. Each acetyl-CoA feeds into the Krebs cycle. So essentially you have two Krebs. After this point here, you have two of everything, two Krebs cycles. This Krebs cycle. Krebs cycle. So that's very important to understand in the first place, that everything happens twice after glycolysis because you have two pyruvate. Okay, so on we go from there. In the previous video, I got as far as going through the Krebs cycle, and at the finish of the Krebs cycle, then I said that by that stage, we had worked out how many ATPs had been produced. Remember, at the start, I said we were looking for a total of 38 ATPs. 38 ATPs. So when glucose is completely broken down through all these stages here, in aerobic respiration, you should get 38 ATPs. But by the time you get to the Krebs cycle, really all you've got is you've just got two ATPs from glycolysis. You get two ATPs from glycolysis, and from the Krebs cycle, from each turn of the Krebs cycle, you got an ATP. So you got another ATP here, one ATP, and from this other turn, you get another ATP, so another one ATP. So by this stage, by the time you get to the end of the Krebs cycle, you've only got a total of 2 and 2. So you've got 4 ATPs so far. 4 ATPs. Now, that's not very good at this stage because you're hoping to get 38 ATPs in total. But by this stage, you've only got 2 ATPs. So where do the other 34 ATPs come from? Well, essentially they come from the electron transport chain, and it's it that I'm going to try and look at now. The electron transport chain is a series of reactions that involve the hydrogen atoms that were released at all these different stages. And if you remember, at all these stages in glycolysis, in the link reaction, and the Krebs cycle, there was lots of occasions where hydrogen atoms were released, and those hydrogen atoms were taken up or picked up by molecules, by hydrogen carrier molecules. And the most famous of those hydrogen carrier molecules is NAD. occasionally there's a couple of situations where you have an alternative hydrogen carrying molecule called FAD, and that occurs in the Krebs cycle. But essentially, the hydrogens that are released in all these steps are picked up by NAD or FAD. And these NAD or FAD are really just delivery molecules. They pick up the hydrogens from these stages and they deliver the hydrogens to another reaction. And the other reaction, or another set of reactions that they deliver them to, is called the electron transport chain. So it's the electron transport chain essentially that we want to be looking at in this video, electron transport chain. So I'm going to move on here and try and explain the electron transport chain. Now, electron transport chain is difficult to draw and quite difficult to explain. So I probably won't draw it on the board at this stage. It's a bit too complicated, but I refer you to my notes here, to page 10 in the notes, and there is a pretty good diagram there. It's always difficult to get a simplified diagram, but that diagram is as simplified as we can get. And essentially, in that diagram, usually I would say to the student, up at the top left-hand corner, up here at the top of the page, I write the word start, because that's the kind of the starting point for these series of reactions, the starting point of the electron transport chain, up in the top left-hand corner. So I'm going to make an attempt to do a very simplified version of it. So if we imagine up in the top left-hand corner, you'll see NAD there, NAD, and NADH. Okay. And you'll see that they're linked with arrows like this. Okay. There's arrows like that. And essentially what I would indicate here is that I would usually put an arrow like this. And I would say, you see that NADH there? That's an NAD. Well, I'll call that reduced NAD. You can call it reduced NAD, NADH. Yeah, reduced NAD is pretty good. Where does that reduced NAD come from? And the answer is, it could come from any of those places in glycolysis, or the link reaction, or the Krebs cycle, where you saw NAD picking up hydrogens. So there's one of the NADs that picked up some hydrogens way back, say, in glycolysis, and there he's arrived. That NAD has arrived there with its hydrogens. Now, what does it do with its hydrogens? Well, you'll sort of see in that diagram, and there's a next stage then where you have something called flavoprotein. And I'm just going to indicate flavoprotein with, I'm just going to call flavoprotein FP, flavoprotein. And then down below, well, up the top you have reduced flavoprotein, reduced. As I say, this is really difficult to draw. You probably need to take the whole screen here, the whole board to draw it. And I'm not going to draw the whole thing because it's just I wouldn't get anything done this hour if I spent the whole time doing. I'll come to my drawing. My drawing on the top of page 10 is very good and really all you need. So you've reduced FAD, sorry, FP, and down the bottom you have FP, flavoprotein. And you'll see that they are linked with arrows as well. They got flavoprotein and reduced flavoprotein linked. Now, really, I want to draw those two just to say this. There's the NAD that's arriving with its hydrogen, reduced NAD. What does that reduced NAD do with its hydrogen? And the answer is, it gives its hydrogen away. It passes its hydrogen on to the next hydrogen carrier. And the next hydrogen carrier in the electron transport chain is this flavoprotein. And you see there's the flavoprotein. There's the hydrogen coming from the NAD. And it's been given to flavoprotein. When flavoprotein gets the hydrogen or gains the hydrogen, it becomes reduced. Because when a molecule gains hydrogen, it's said to be reduced. So you go from flavoprotein to reduced flavoprotein. So essentially the flavoprotein has taken the hydrogen off the NAD And in so doing, it's now reduced flavoprotein. What has happened to the NAD? Well, the NAD has lost its hydrogen. It's now back in the oxidized form. It's now NAD. It's gone back to NAD. What happens to that NAD? That NAD simply goes away back again to glycolysis or the link reaction or the Krebs cycle and picks up more hydrogens, picks up another hydrogen and brings it back here. So really all the NAD is a delivery molecule. It just goes away and picks up some more hydrogen atoms and delivers them here to, we're now looking at the electron transport chain. Okay, and so it goes away and gets some more hydrogens and delivers them to the electron transport chain. So NAD hitch, that's reduced NAD, yeah, and there's oxidized NAD. So oxidized NAD goes away back and gets some more hydrogens and becomes reduced again. Now, we're going to just look at this step for a wee second, because you'll see something else in this step. You'll see indicated on this step that there is some energy released. So this reaction step releases some energy. Energy is released, ERGY is released in this step. And that energy is used to make an ATP molecule. Because the reaction to make an ATP molecule involves ADP plus P mix ATP. And this reaction needs an input of energy. Where does that energy come from? It comes from this reaction, because this reaction releases some energy. So this step releases energy, and that energy is used to make the ATP, because the making of ATP needs energy. So where is the energy gone? The energy is now stored in the ATP molecule. So you've made an ATP molecule in this case. Now, this step here, if I was to write this step as a reaction, I'm going to rub it out now. I'm going to write it more as sort of like this. Here's what we would have. Basically what it would look like is this. So what did we start with? We had NADH, yep, and it reacted with, or somehow or other, reacted with flavoprotein, FP, okay, and what was made? FPH, in other words, reduced flavoprotein, plus the NAD, and this reaction releases energy. Yeah. So one reaction, this reaction releases energy, and that energy is used to make an ATP. Yeah. So this would be described as a chemistry would call this an exothermic reaction or an exergonic reaction. A reaction releases energy, and this, of course, is an endothermic or endergonic reaction that needs energy for it to happen. So you can see what's happening. You've got the coupling of this reaction and this reaction. And the end result is you've made an ATP. So there you are. So that reaction up there involved just the flavoprotein taking the hydrogen from the NAD. And that's the first step. And you get your first ATP there. And then you can see from your electron transport chain what happens next. Your flavoprotein, your reduced flavoprotein then passes its hydrogen on to the next carrier, and the next carrier is something called Coenzyme Q. Coenzyme Q. And really what the electron transport chain is just a series of carriers from the start to the finish. A series of carriers. And the ones that are obvious there, so the carriers are flavoprotein, coenzyme Q, reduced cytochromes. There's actually 2 or 3 of those, but you don't need to worry about the fact there's 2 or 3 cytochromes. You can just regard them as cytochromes. And then there's something called cytochrome oxidase. And then eventually, and very importantly, the very last carrier in the sequence is oxygen. And I've highlighted oxygen here. Oxygen. So really, The electron transport chain is just a series of steps along which hydrogen is passed, and those steps involve the release of energy. But it's a wee bit more complicated, just a wee bit more complicated than that. And usually at this stage, students are getting confused a bit, or they're getting fed up with this. And so I say to them this: you can learn all the steps, or you can think about, you can look, if you look at my notes, you'll see that I've got about 9 steps for the electron transport chain. Or you can do this. If you look at questions to do with the electron transport chain, and I'll just bring you to a typical example of one. And this is quite a common question, just about the electron transport chain. So in my booklet there, you'll see question 10, the second part, part 4 of question 10. So you go to part 4 of question 10, and essentially part 4 of question 10 says, explain how ATP is produced in the electron transport chain. And you see they give you 4 marks for doing that, and give you about 10 lines to write. That's the typical way in which they ask about the electron transport chain. They simply say something along those lines. Tell us, how does the electron transport chain work to make ATPs? And they give you 4 marks. And for 4 marks, you need 4 points. So you may think, well, I've got about 9 points there explaining it. Well, my suggestion would be this: even though they're only asking for 4 points in their question, I would squeeze in as much of that information as possible into their 4 points. Or alternatively, if you look up on the top right-hand corner of page 10 there, you'll see that I've written, there's a little thing that I've said, this is the answer scheme for a typical question. Actually, that's the answer, there's the answer scheme for that question we've just looked at. And that's the answer scheme that the examiners would want. And they basically want, how many of those points? There's, in that answer scheme, there's 6 points. They want any 4 of those. So actually it's quite generous. So as a sort of shortcut for the electron transport chain, you could learn that, you learn those 6 points, or you learn the 4 most important ones. I wouldn't recommend that. I'd recommend learning them all 6 points. In fact, my best recommendation is to know it all, because that just gives you a better understanding. So in the first reaction, then, NAD gave its hydrogens to flavoprotein. Flavoprotein was reduced. And you'll see in the second reaction, the flavoprotein gave its hydrogens to coenzyme Q. In so doing, the flavoprotein and the reduced flavoprotein became flavoprotein again. It was oxidized because it gave away its hydrogens. What happens to the coenzyme Q that gains the hydrogens? It's now said to be reduced coenzyme Q. So there's a mark here in the these points here for saying these are a series of oxidation reduction reactions that's happening in the electron transport chain. A series of oxidation reduction reactions. Some schools would say a series of redox reactions. It's the same thing. A series of oxidation reduction reactions. In other words, a series of reactions in which one molecule is oxidized while the other is reduced. And a molecule is reduced when it gains hydrogen, and it's oxidized when it loses the hydrogen. Again, but the slight complication here, after the third carrier there, coenzyme Q, what happens is that the hydrogens that have been passed from carrier to carrier, hydrogens split up into hydrogen ions and electrons. In fact, a hydrogen ion, which is just a proton, and an electron. So the hydrogen actually splits in 2, and it's actually just the electrons that are passed then through the carriers from there on. So it's just electrons that are passed. So coenzyme Q passes electrons to the cytochromes, and then the cytochromes pass the electrons on to cytochrome oxidase, and so on. So it's electrons. Now there's a mark in the mark scheme for that. You'll see in the mark scheme, I'm just reading through the 6 points that are here in the typical mark scheme, and what they wanted you to do was first of all say hydrogen enters the electron transport chain. So you could say hydrogen comes into the electron transport chain as NAD, as reduced NAD, And then the second mark, hydrogen atoms pass initially, and then electrons passing thereafter. What they want you to say is initially, hydrogen atoms are passed from carrier to carrier, and then later on it's electrons that are passed from carrier to carrier. As long as you say that, you get marks for that. And then the third mark there is quite nice. It says one carrier is oxidized as hydrogen or electrons are removed. And the next carrier is reduced whenever hydrogen or electrons are gained. So just something to notice there, that if a carrier gains hydrogen, it's reduced. Equally, if it gains electrons, it's reduced. If a carrier loses hydrogens, it's said to be oxidized. If a carrier loses electrons, it's said to be oxidized. Okay, so you would get that mark. Now, you'll notice that, that the electron transport chain isn't in a straight line, it runs downhill. And the downhill idea is this bit like in physics, potential energy. As you go from carrier to carrier, each carrier has less and less energy. The energy is being lost as you move from carrier to carrier. And so you get this downslope. And that there is encompassed in the fourth bullet point in the mark scheme. The carriers are at successively lower energy levels. So that's a good one to say. Students always say that carriers are at progressively lower energy levels. So as you move from carrier to carrier, there's less and less energy in the system. And that energy is being released at different stages. Now, you'll notice energy was released in the first step. But there was no energy released in the step that went from flavoprotein to coenzyme Q. No energy released in that step. But then in the next step, between coenzyme Q and reduced cytochromes, energy is released again. So that reaction in which reduced coenzyme Q passes electrons to one of the cytochromes, making that cytochrome reduced. That step is a energy releasing reaction, and that energy is used to make your second ATP. So you see your second ATP being made there. So there's your first, here's your second ATP. And then you'll notice in the step between reduced cytochromes and cytochrome oxidase, when electrons are passed there, there's more energy released. So notice then that there are 1, 2, 3 ATPs. In other words, 3 points where energy is released, enough to make 3 ATPs every time you get an NADH arriving at the electron transport chain. So every NADH is worth 3 ATPs. Okay, so bear that in mind. Every NAD that delivers hydrogen to the electron transport chain, that results eventually in 3 ATPs being made. 3 ATPs being made. Now, just to notice when you're there, you'll notice FAD, and if you remember, FAD was a carrier in the Krebs cycle, and there it's up at the top here, kind of sticking out a bit, kind of in the road a bit. But basically, you notice that FAD delivers its hydrogen to coenzyme Q. FAD misses out the first step, and therefore you only get 2 ATPs made every time FAD delivers hydrogen to the electron transport chain. So for every FAD, you only get 2 ATPs. How many FADs deliver hydrogen to the electron transport chain for every molecule of glucose? Answer is just 2. Remember, there was an FAD. There's one FAD in the Krebs cycle, and the Krebs cycle happens twice. So it's really 2 FADs. And those 2 FADs, each of them end up producing 2 ATPs. So those 2 FADs total produce 4 ATPs. Yeah. Okay. So what else is to say there? And you'll see that that's another mark in the mark scheme. You'll see 3 this is the very last bullet. It says 3 ATPs produced when you have reduced NAD, and 2 ATPs from reduced FAD. Yeah. The second last point said sufficient energy is available at certain stages. You could have said released at certain stages to produce ATP. At certain stages is important there. I would underline that in the mark scheme, because you really let an examiner know that you realize energy isn't released in every step, it's just released at some steps. So there you are. There's the 6. There's the summary of the electron transport chain in 6 steps. If you're worried about it, I would simply learn that mark scheme, because that mark scheme is a sort of a summarized version of my 9 points. Yeah. So if you look through my 9 points, essentially I've set those 6 points, but I've maybe just elaborated a wee bit on each of them. Now, it's useful to look at point 9 on my notes there. And I said, in the very final step in the electron transport chain, the hydrogen ions combine with the electrons. Remember we said earlier that the hydrogen ions split into, the hydrogen split into hydrogen ion and electrons. Well, they recombine again in the very last step. So the hydrogen ions get back with the electrons to make hydrogen. And the cytochrome oxidase then passes the hydrogen ions and the electrons together, and they are given to the final acceptor in the sequence. And who is the final acceptor in the sequence? Not made obvious enough there, but I would make it very obvious. Oxygen. Oxygen is the last in the sequence of carriers. Oxygen's there at the end of the sequence. Oxygen's there, and the oxygen takes the hydrogen. So you have hydrogen and oxygen, and that makes water. Oxygen's often referred to as the final hydrogen acceptor in the electron transport chain. And so I often say to students, this is a bit like a game called Pass the Partial. I don't know what this game involves, but Pass the Partial, I imagine, is a game where you have a partial or a package and you pass it to your friend, and they pass it on and pass it on and pass it on to the very end. This is like a game like that. And at the very end of the sequence, you've got oxygen. Oxygen's the last stage in the sequence, and oxygen accepts the hydrogens, and in so doing becomes water. Now, why is oxygen important? Well, it's important to allow the electron transport chain to happen. And it's important, you notice its importance when it's not there. When oxygen's not there, oxygen's not available at the end of the sequence of carriers. There's nothing to accept the hydrogens. The hydrogens aren't accepted. The last carrier in the chain can't pass the hydrogens on to oxygen. And so what happens is that all the carriers in the sequence end up holding hydrogen, because any new NADs that arrive at the chain with their hydrogens need to pass their hydrogens on to flavoprotein. But if there was no oxygen down at the end of the chain, then all the carriers in the chain end up holding hydrogen. They can't get rid of their hydrogen. Why can they not get rid of their hydrogen? Because the next carrier can't get rid of their hydrogen. Because the next carrier can't get rid of their hydrogen. Why not? Because oxygen's not there at the end of the chain to accept the hydrogens. So oxygen is absolutely vital in this process. Oxygen acts as the final hydrogen carrier, final hydrogen acceptor, and becomes water. And that last carrier is actually an enzyme, cytochrome oxidase. And if you read about it at the very bottom of the page here, cytochrome oxidase enzyme, step 9. In the last stage, hydrogen ions combine with electrons to make hydrogen. The hydrogen then combines with oxygen to make water. The enzyme cytochrome oxidase catalyzes this last step. So there's an enzyme here, and it catalyzes this last step. Now, this enzyme is very important. Cytochrome oxidase enzyme can be inhibited by a respiratory poison called cyanide. So you can inhibit, and you remember back to AS1, where you looked at how enzymes could be inhibited. Well, this is an example of non-competitive inhibition, where the cyanide, a molecule called cyanide, can combine with the enzyme and block the active site permanently, or can block the active site permanently, or distort the shape of the enzyme permanently so that the enzyme doesn't work anymore. So this cyanide is a very dangerous molecule because it stops respiration anywhere where it comes across respiration. If this happens, hydrogens cannot combine with oxygen to form water. The hydrogen builds up in the electron transport chain, and all the carriers remain reduced. So if this enzyme can be inhibited, then you can stop respiration. And you might say, when does this happen? Well, it happens when the cyanide stuff's around, and this cyanide is recognized as a very dangerous poison. There are lots of instances where you come across people being poisoned with cyanide accidentally or otherwise, and it usually means death very quickly. Cyanide gets into your body, it gets to all your cells, it inhibits this enzyme in every cell. It stops then the electron transport chain because it stops the last step in it. If you can stop the last step, the whole thing backs up. If you stop the electron transport chain, then you stop the release of all these ATPs that are released in the electron transport chain, and essentially then cells cannot release ATP, and the cell doesn't have ATP and it can't do all the processes that cells need to do. Especially, for example, muscle cells need lots of ATP for muscle contraction, and your heart, if you think about it, is a very important muscle, and if cyanide gets into your body, usually you die almost immediately from a heart attack because your heart muscle cells can't do respiration, and yeah, you die fairly quickly. So that's a question about that's the electron transport chain that I covered there. Now, if you look through the booklet of questions there, you'll see that the electron transport chain then is over the years in different questions. It's been asked on that sort of basis. They simply ask you to describe what's happening in the electron transport chain, and usually give you about 4 marks, and you have to come up with the key points there. Now, so that's the electron transport chain. The best thing for you to do is to actually have a go at doing the questions there. I'm just going to look through the whole question booklet here and look at instances where you come across questions about the electron transport chain. So in question, just looking for the questions there. Yeah, question 4 on page 6. Part C, a very old question, says, what happens during the electron transport chain resulting in the production of ATP? There's 4 marks there. It's a very old question, and notice there's only about 6 lines. A modern day question would give you about 10 lines and give you the 4 marks. So there's one example of it. Turn the page to question 5, the bottom of page 5, question 5. The bottom of the page, page 7. Explain how the respiratory chain operates to generate ATP from reduced coenzymes NADH and FAD. Now, that's just another way of asking about the electron transport chain. And once again, you get 4 marks there. Now, part D on page 8. Explain the production of 11 ATPs in the respiratory chain. By the way, respiratory chain is just another name for the electron transport chain. So you can write about respiratory chain, electron transport chain. And it says in this particular question, explain the production of 11 ATPs from the electron transport chain from 1 turn of cycle A. But if you look on that question 5, cycle A is the Krebs cycle, isn't it? So let's just think about the Krebs cycle then. 1 turn of the Krebs cycle, how many ATPs do you get from that? From the electron transport chain. You'll remember that in 1 turn of the Krebs cycle gives you 1 ATP directly. So you get an ATP anyway. But that's not what this question's asking. It's saying how many ATPs do you get from the electron transport chain as a result of 1 turn of the Krebs cycle. So you have to think about the Krebs cycle and think how many reduced NADs and reduced FADs did you get from the Krebs cycle? So if you remember, the Krebs cycle gave you 3 reduced NADs. Remember, there were 3 occasions where you get to hydrogen released, 3 reduced NADs and 1 reduced FAD. Okay, so you have 3 reduced. So each turn of the Krebs cycle give you 3 NAD hitches and 1 FAD hitch. Now, what have we just discovered? We've just discovered that every time you get an NAD hitch arriving, these go to the electron transport chain. Every time you get one of these arriving, an NAD hitch, you get 3 ATPs. So 3 of them arriving would give you 3 by 3 is 9 ATPs. So you get 9 ATPs from the 3 NAD hitches, from 3 reduced NADs that go to the electron trap. And the 1 FAD just gives you 2 ATPs. So you get a further 2 ATPs. And so you add those together, you get 11 ATPs. If that answers your question. So one turn of the Krebs cycle gives you 11 ATPs. And essentially, to answer that question for 2 marks, you'd say in each turn of the Krebs cycle there are 3 reduced NADs, 3 NAD hitches released, and 1 reduced FAD. You can call it reduced FAD, or you can call it FAD hitch 2. And then you could say each NAD hitch, each reduced NAD generates 3 ATPs of the electron transport chain. Each FAD generates 2 ATPs, and then 3 NAD hitches will give you 9 ATPs. 3 NADs will give you 9 ATPs. 1 FAD will give you 2 ATPs. 9 plus 2 is 11. I should probably give you more marks there for that. There's quite a lot of explaining. So maybe at this point, then it's useful to try and go back through the glycolysis and the link reaction and all those, and try and count how many ATPs you get in total then from all the NADs that are released in all the different stages. So I'm just going to try and come up with a sort of a table for that. And I don't actually have a table in the notes there, but we'll have a quick think about that. So what are we asking? We're asking how many ATPs in total do you get from the electron transport chain for every glucose that goes through respiration. So we'll just do that quickly again. So glucose, and each glucose then is converted into 2 pyruvates. There we go. And then each pyruvate is converted to acetyl-CoA, CoA. And each pyruvate acetyl-CoA, CET-CoA. And then each of those goes into a Krebs cycle. Okay, now this is going to be pretty rough, but here we go. So glycolysis. Going back to glycolysis, how many reduced NADs were there? Well, if you go back and look in your notes at glycolysis, you'll see that there was just 2 reduced NADs. So back in glycolysis, there's just 2 reduced NADs. I'm just trying to remember where exactly the steps are. So 2 reduced NADs. I'm looking back at page 6 in the notes here. So glycolysis gives us 2 NAD hitches. Okay, all right. And then the link reaction. Well, the link reaction happens twice, and each step in the link reaction also gives us an NAD hitch. So in the link reaction, then you also get 2 NAD hitches in total. Isn't that right? 2 NAD hitches from the link reaction. And then the Krebs cycle gives you how many NAD hitches? Well, each turn of it gives you 3. So you get 3 NAD hitches. But the 2 turns of it then would give you a total of 6 NAD hitches. Okay, all right. So 3 NAD hitches from each turn in the Krebs cycle. Oh, and as well as that, then you got how many FAD hitches? Many reduced FAD? You get 2 of those. 2 FAD hitches. So if you want to count up in total, then the total number of reduced NADs was 2, and 2 is 4, and 6 is 10. So you get a total of 10 NAD hitches, and you get a total of 2 FAD hitches. And then if you remember, each NAD hitch that arrives at the electron transport chain gives you 3 ATPs. So 10 NAD hitches would give you 30 ATPs. and the two FADs, each of them gives you 2 ATPs. That would give you 4 ATPs. So there you are. The total ATPs coming from the electron transfer chain are 34 ATPs. So there you are. You add them all up now. So 34 ATPs from the electron transfer chain, and the other 4 just came from what's referred to as you can refer to it as substrate level phosphorylation. And if you remember the other ATPs, you got a little ATP made in each turn of the cycle here, of the Krebs cycle. You get a little ATP there. That was 2. And then you remember glycolysis made you 2. Glycolysis made you 2 ATPs. So you have 34 ATPs from the electron transport chain, and then you have a further 4, which gives you 34 and 4 is 38. So there's your 38 ATPs. So often they'll ask questions like that. Often they'll ask a question that says, comment on the amount of ATP produced in the different stages of the electron of respiration of glucose. And one comment you could say is most of the ATP comes from the electron transport chain. Only a small amount comes from what you call substrate level phosphorylation. By the way, the electron transport chain and the events that are often referred to as oxidative phosphorylation. So the electron transport chain, oxidative phosphorylation. These other ATPs produced here and here, substrate level phosphorylation, oxidative phosphorylation, and substrate level phosphorylation. Now, just looking to see sorts of questions they ask about this. Just leafing through the questions again. So I'm going through question 5. Question 6 is on anaerobic respiration. We'll come to that in a moment or two. Question 7. Question 7, just at the bottom of question 7, 7C at the bottom of the page, bottom of page 10, you can see once again it's another electron transport question. Electron transport chain question says, how does the respiratory chain, that's the electron transport chain, use hydrogen and mix ATP? Once again, 4 marks, and you can see there's your 4, you're going to get your 4 bullet points there. Question 8 is looking at glycolysis, and that question 9 doesn't ask us about the electron transfer chain. Question 10, then you'll see 10 part 4 is the question I showed you at the start with the electron transport chain. A typical question with 4 marks. And I think that's possibly the total of those questions, total of the questions on the electron transfer chain, except to say that question 12, which is a very old question, gives a very good diagram of the whole process. And it kind of summarizes it quite well. A lot of what happens in the process of respiration. So I like question 12. It asks you, it makes you look at the whole process. Okay. Right. Good. So that's the electron transport chain. Not easy, but not impossible. As I say, your best shot is to first of all, for the first thing, is learn a series of bullets there for that 4 marker question. Know that, because that's the most commonly the kind of question you get asked. By all means, go through. By all means, look at where all the NADs are produced in the whole respiration of the whole breakdown of glucose, where the NADs and FADs are. And you can draw yourself a simplified version of the electron transport chain, but take my word for it, you'll never be asked to draw the stages in the electron transport chain. You'll not be asked to name the carriers in it. You'll not be asked to do any diagrams, complete any. Only question I've ever seen is essentially those 4 marker questions, where it simply asks you give a description of the electron transport chain. So it becomes not an impossible question then. Now, I'm going to briefly look at anaerobic respiration. Now, in talking about anaerobic respiration, we need to talk about the importance of oxygen in respiration. So just going back to the electron transport chain there, I was trying to point out very clearly that the importance of oxygen at the very end. Oxygen, the last carrier, the last hydrogen acceptor. Essentially, if oxygen is not there, if oxygen is there, everything happens perfectly. The glucose gets completely respired, completely broken down. You get all the stages of glycolysis. Sorry, you get all the stages of respiration, glycolysis, the link reaction, Krebs cycle, and all those stages happen perfectly. But the importance of oxygen is only pointed mid-obvious once there's no oxygen there, once the oxygen's not there. So I refer you to page 11 in the notes here, where I've kind of spelt out very clear, spent a lot of time there talking about the importance of oxygen. So I'm just going to essentially read through these points so that I don't miss any. I've said when a cell has, you can underline has, oxygen available to do respiration. This is known as aerobic respiration. In aerobic respiration, all the stages of respiration take place. So in aerobic respiration, you get glycolysis, the link reaction, Krebs cycle, and the electron transport chain. And you get a total of 38 ATPs mid for every glucose that you start with. And I've asked the question, where precisely is oxygen needed in aerobic respiration? And the answer is, it's needed as the final hydrogen acceptor in the electron transport chain. And I've said then, the electron transport chain can only happen if oxygen is present, and I've highlighted that statement. And then I've kind of summarized why the electron transport chain. I've said NAD or FAD arrives at the electron transport chain in the reduced state, i.e. bringing hydrogen or carrying hydrogen. NAD or FAD pass the hydrogen along the carriers until it reaches oxygen eventually, and oxygen is the final acceptor. Oxygen combines with the hydrogen to create water. And then the last point, which I've asterisked, I've said oxygen needs to be available. It must be there to accept the hydrogens, which pass down the electron transport chain along the carriers. So the oxygen must be there. Now, if oxygen is there, everything works well. And then I've asked this question, what happens to the NAD that delivered the hydrogen to the electron transport chain in the first place? And the answer is, that NAD goes back and collects more hydrogens, just returns to wherever and collects more hydrogens from glycolysis, a link reaction, Krebs cycle. So you've lots. So the important thing is, if the cell has got lots of these NAD molecules available to it, and basically they're just delivery molecules. They're collecting hydrogens and delivering them to electron transport chain. So the NAD is a very important carrier. So I've said at the top, page 12, NAD is simply a carrier molecule or a delivery molecule which collects hydrogens and delivers them to electron transport chain. Now I've asked a very important question. I've said, what happens at the electron transport chain if there's no oxygen available? If there's no oxygen there. In other words, if oxygen hasn't arrived to the cell, if the cell doesn't have oxygen inside it, it's not getting oxygen delivered, then I've said there's no oxygen to accept the hydrogens at the end of the electron transport chain. Oh, so there's no oxygen there. What now happens? Essentially, the electron transport chain stops. Why does it stop? Well, I've said if there's no oxygen, then the carriers in the electron transport chain now all remain in the reduced state. That simply means that NAD arrives with its hydrogen, passes it to flavoprotein, passes it to coenzyme Q, it passes it on to this, eventually the hydrogen passes through the cytochromes, but then oxygen's not there to take the hydrogen. So basically, the hydrogen that's been carried by cytochrome oxidase at the very end, it can't get rid of its hydrogen. So all the other carriers back up, they all end up holding hydrogens that they can't get rid of because oxygen hasn't turned in. Oxygen's not available. So if there's no oxygen, then the hydrogens can't pass along the electron transport chain. So what happens to the NADs that arrive at the electron transport chain with hydrogen? The answer is they can't get rid of it. They can't give their hydrogen to flavoprotein because flavoprotein is already holding a hydrogen because it can't get rid of its hydrogen. So NAD arriving at the electron transport chain, if there's no oxygen there, The NAD simply is left holding the hydrogen. The NAD is said to be left in the reduced state. It can't get rid of its hydrogen. And that means that eventually, inside the cell, if the cell doesn't have oxygen, all the NADs in the cell all pick up hydrogens from various places, but they all arrive at the electron transport chain and it's not working. So basically all the NADs in the cell end up holding hydrogen. They all end up in the reduced state. So there's no NADs being made available to go back and collect more hydrogens. So that means that the steps in the glycolysis, the link reaction, and the Krebs cycle that required NAD to be present to collect hydrogens, those steps won't happen. So if the NAD is not there, all those steps in the back-end respiration don't happen. So essentially, all the steps in glycolysis, the link reaction, and the Krebs cycle, that all grinds to a halt as well. Everything grinds to a halt. So once the electron transport chain doesn't work, the rest, all the other stages don't work either. And the cell doesn't have oxygen. So glycolysis and the link reaction, the Krebs cycle all stop. But there's some hope for the cell, because you'll see at the bottom of page 12 I've said, what happens when the cell isn't able to continue doing aerobic respiration? In other words, when the cell has no oxygen. And the answer is, the cell can revert or can do a different kind of respiration called anaerobic respiration, which is like a kind of second class kind of respiration that allows the cell to make some ATPs, but not very many. In fact, it allows the cell just to make 2 ATPs. Remember, aerobic respiration made 38 ATPs. This alternative anaerobic respiration only allows the cell to make 2 ATPs. And essentially, this is what we want to look at. How does the cell, what happens in anaerobic respiration? Well, just there I've said that if the cell doesn't have oxygen, then all the stages in respiration stop. Glycolysis, the link reaction, and the Krebs cycle, and the electron transport chain. But I've said that the cell gets 2 ATPs. Now, where does it get the 2 ATPs from? Well, actually what happens is that cells have developed a way of getting 2 ATPs even when there's no oxygen. Now 2 ATPs is not very good, but at least it's some ATPs. And the answer, how does that happen? And the answer essentially is that the cell is still able to do glycolysis. The cell has found a way of doing glycolysis, even though it's got it doesn't have NAD to collect the hydrogens in one of the steps in glycolysis. And if you look in glycolysis, you'll see there glycolysis and the step that goes from triose phosphate to glycerate bisphosphate. You'll see you need NAD to be present there to make that step happen, to allow that step to happen. So there is no NAD available. So how does the cell get to do glycolysis if it's got no NAD? Well, this is just briefly, and I think I might have to go into this in a further video because I don't want to rush it too much. But essentially what happens is that this is glycolysis. You start with glucose. And I'm not going to draw all the intermediate steps. I'm simply going to say that in glycolysis you go from glucose to pyruvate. And one of those steps needs NAD to be present to collect hydrogen. So it's a step from triose phosphate to glycerate phosphate. So you actually need NAD to be there. And the NAD picks up some hydrogen and becomes NADH, or just NAD. But we're saying this NAD is not available. It's not available. Why is it not available? Because it's away at the electron transport chain with hydrogen, and it hasn't come back because it couldn't get rid of its hydrogen. So NAD is not available. So how does the cell get around this, the need for NAD? So there's no NAD available. What does the cell do? And well, it turns out that cells have developed an extra reaction step. And basically the extra reaction step involves the pyruvate being converted into another molecule called lactate or lactic acid. Lactate. And here's essentially what happens. Essentially, this is the way I usually explain to students. I say that if we could get glycolysis to happen, we can get 2 ATPs. Okay, the cell has found a way to do this, and here's how it does it. What happens is that pyruvate acts as a hydrogen acceptor. Pyruvate acts as a hydrogen acceptor. Essentially what happens is pyruvate sort of steps forward and it accepts the hydrogen from NADH. So basically NADH gives its hydrogen to pyruvate. And pyruvate accepts the hydrogen. And when pyruvate accepts the hydrogen, that means the NAD is freed up to do this step. So how you would write this is you say pyruvate, you write it like this, NADH becomes lactate plus NAD. So essentially the pyruvate has taken the hydrogen from the NADH. And so doing, the pyruvate becomes a new acid called lactic acid or lactate, but it has freed up the NAD. And once that NAD is available, that NAD can go up here and do this step. It can take off more hydrogens. And that means glycolysis can continue to happen. If glycolysis can continue to happen, you get 2 ATPs. You get 2 ATPs. And so the way it's often illustrated would be like I have in my notes there. If you look on page 13, and we'll just read down through page 13. Page 13, anaerobic respiration. When a cell has no oxygen, the electron transport chain cannot happen, and no NAD is regenerated. That means there's no NAD freed up to come and collect more hydrogens. No NAD is available to return to the reaction to glycolysis link reaction Krebs cycle to collect hydrogens. However, the cell is still able to do glycolysis, and this involves an extra reaction step. And the extra reaction step is where pyruvate accepts the hydrogens from NADH, and in so doing it becomes lactate, but it frees up the NAD. So essentially the reaction is pyruvate takes the hydrogen from NAD and it frees up the NAD, and in so doing it makes another molecule called lactate, and the NAD that's been freed up then can make glycolysis continue because it can keep taking off more hydrogens, and those hydrogens react with pyruvate, and the whole thing just keeps going on. So now, how is this asked? Well, you'll see question 1 there, a very recent question, and you can see question 1 is basically about anaerobic respiration. First of all, it asks an interesting question. It says, where does this happen? Where does anaerobic respiration happen? Well, anaerobic respiration is essentially just glycolysis plus this extra reaction. So glycolysis happens in the cytoplasm. So the answer is the cytoplasm. It says, energy release in anaerobic respiration is a rapid process. And well, the reason it's rapid is it just involves a very short part of the whole glycolysis, of the whole respiration process. So it just involves a very short part. In other words, glycolysis plus this extra step. So just a short part of the whole process. And then it says, explain the importance of A. Well, essentially this is the nuts and bolts of it. A is basically the stage where pyruvate becomes lactate. You could say it's this step. So basically the importance of this step is that the pyruvate is acting as a hydrogen acceptor. Sometimes it's said to be acting as an alternative hydrogen acceptor. Acting as a hydrogen acceptor, accepting hydrogens from reduced NAD, releasing the NAD so that the NAD can continue to remove hydrogens during glycolysis. So essentially that's the answer you'd say. Important to this step is that pyruvate is accepting hydrogens from NAD. It's acting as a hydrogen acceptor, freeing up NAD or regenerating NAD is often said. You'll see in my notes there, I said in this reaction step, pyruvate acts as a hydrogen acceptor. Pyruvate accepts hydrogens from reduced NAD. This frees up or regenerates NAD. And the regenerated NAD now is available to continue removing hydrogens during glycolysis. And glycolysis can now continue and the cell can now make ATPs. When the pyruvate accepts the hydrogens, it becomes a new acid called lactate or lactic acid. And you'll see down there on the bottom left hand side, you see essentially that diagram. That's what happens in so this is what happens in humans, animals, and bacteria. This is the way anaerobic respiration happens in animals and bacteria. But in plants, it's slightly different. You'll see there in plants, the pyruvate accepts the hydrogens. Instead of becoming lactic acid, it becomes something called ethanol, and the CO2 is released. So its reaction would be pyruvate plus NADH becomes ethanol plus CO2 plus NAD. It's essentially, so it's a very similar process, except you've got ethanol and CO2. And in the vast derivation, I give two differences between the process of anaerobic respiration in plants and animals. You'll see at the bottom of 13 there. You'll see there's a couple of differences then. Okay. The most recent questions asked about this are about anaerobic respiration. I'm going to stop just now. Is that question one? That's a very recent question. I think it's 2018. And so the first two parts of it, part B, I'm just going to do very quickly. It says, suggest why many organisms favor aerobic over anaerobic respiration. Why do organisms prefer aerobic? The answer is you get more ATPs. And the second mark is there. If you say well, aerobic respiration, you get 38 ATPs, whereas with anaerobic respiration, you just get 2. So I'm going to stop at this stage. If you have my question booklets, then you've got the answers. You should have the answers for the rest of those questions. It's quite a difficult topic, anaerobic respiration, and you can only get good at it by doing the questions. So I may at some future time do another video on this. This would be do the third video on respiration, whereby I go through all of these questions and talk you through all of them. You have the answers for them already, but I could talk you through all of these questions. Okay, that's as much respiration as I'm going to do today. So there you go. All the best then. Thank you very much. Bye.